everyone, welcome back to the cell signaling course. We have been discussing receptor serine threonine kinase in different contexts. In case of plants, we saw how it is important for every aspect of plant development. And in case of receptor serine threonine kinase signaling in animals, we have been seeing that it plays very important role in bone morphogenesis as well as lot of early developmental effects. Today in the class, we will see that how very specific signaling which is mediated by activin kind of molecule and an anti-Mullerian hormone, they are working through receptor serine threonine kinase to meet the signaling and its responses. So what exactly is this activin signaling is? This is a small molecule which is produced by different cells and they work on very different types of cells. They respond depending on what cells are there and what kind of processes they will be controlling and governing. But lot of immunity based things where activin pathway is activated, and you will see accordingly the response will be mounted, whether it is in context of infection, it is in context of autoimmunity or cancer and so on. So activin, again, as we have been seeing for a regular receptor serine threonine kinase signaling, it is made up of heterotetrameric assembly of two receptors which engage with the ligand. Here we are saying activin A. The proteins or the cells often which make activin A also respond to another molecule which is called inhibin. So basically simultaneously regulating the activin receptor activity. So the two classes of receptor coming into tetrameric assembly follow the canonical pathway where R-SMADs are phosphorylated, they bind with the co-SMAD, go inside the nucleus, regulate the transcription. And as I said, they mediate multiple different pathways, so it could be related to infection, immunity, growth and so on. And as we have seen that the activation of this pathway can be regulated by degradation of R-SMAD using the ubiquitination pathway or inhibition of R-SMAD from binding to the receptor and thus activating the pathway. But there is interesting regulation at the activin level itself, where the activin can be regulated from its binding to the receptor by multiple different ways. One of the way is that it binds to an inhibitor. So an inhibitory protein which is part of the membrane associates with this activin and receptor complex, and thus it will not be able to activate the system. Removal of this inhibitor is very important. This inhibitor must be removed so that activation can happen. Also, inhibin, the other molecule which I told you, which holds very strong similarity with the activin, it is the ligand for the receptor. It will bind to the receptor and bring another molecule like a co-receptor together to ensure that receptor is not available for activin to bind. This seems very similar to a situation where RLP like proteins were binding to the receptor. However, in that case RLPs were not inhibiting the signaling. Here this molecule seems to be inhibiting the signaling by virtue of presence of this inhibin like molecule. Another way is where this molecule is produced but it is isolated, it is sequestered by this inhibitory molecule follistatin. So as long as this follistatin is in complex with the activin, this is not available and thus it cannot serve on the receptor to mediate definite processes. So you see that activin is working through receptor serine threonine kinase and it is following the canonical pathway, but on the other hand you have set of regulation at the level of activin itself or the receptor activation itself. Now let's take example of activin signaling in different pathways. If we think about the infection, so many different cell types in our body which respond to infection, like microglial cell in central nervous system, or monocytes, dendritic cells or macrophages. These are basically the cell types which remove the infection from the system, either by phagocytizing them or killing them by secreting or releasing certain contents inside them. So the microglia and macrophage, these are the cell, they activate the activin signaling when there is a bacterial infection. Particularly in case of microglia, these are central nervous system cells. So when there is a bacterial meningitis, we understand that meninges are the membranes which cover the brain tissue. So if there is an infection of bacteria in the meninges and which induces inflammation, that's meningitis. So this meningitis is a cue for these microglial cells to activate the activin signaling, which will remove the infection. It will put the signal into the system where you will have maybe new transcriptional programming as well as immediate activation of these cells so that they can induce phagocytosis or release content which will take care of the infection. Activin signaling also helps in the parasitic infection, where a case of leishmania which is known, where activin signaling is induced when parasite is around. If you recollect, we saw that how in case of plants, multiple components which are derived from pathogen, whether it was a chitin or a polysaccharide or lipopolysaccharide, or for that matter flagellin, which is component of the bacteria. Similarly, these pathogens also have specific patterns. We also saw something which was like PAMP and DAMP. Pathogen associated molecular patterns and death associated molecular patterns. So how plant was responding. Similarly, in case of animals, through activin signaling, when such signals are present from pathogens or because of the infection, if DAMPs are also activated, this signaling is activated, which ensures that the cell responds accordingly by mounting an antipathogen response or undergoing death and in response to the DAMP. Also, the viruses like human cytomegalovirus, their infection is decreased when there is a system around and you have HCMB infection. The signals which are emanating from HCMB or like interferon, we know that some of the cells in our body, in response to infection by a virus, they secrete these compounds which are called interferon, and these interferon can work through this activin signaling and ensure that infection load is decreases, viral load goes down. So activin signaling is playing very important role in different types of infection. Depending on type of infection, different types of cells are involved. Now let's talk about this activin signaling which is happening in case of allergical responses. So here same activin like molecule, this is the activin A. For allergical responses, it is dependent on specific cell types, T cell types, granulocytes, mast cells, dendritic cells and macrophages. What is understood and known that T helper cells of distinct classes, there are different types of cell, T cells, T cell which are cytotoxic, and then there are T cells which are helper cells. So cytotoxic T cells, they will directly be involved in clearance of the pathogen or allergen, but if you have T helper cells, they will be mediating the anti-allergic responses. So when activin signaling is enhanced, it may, depending on the allergen type and cell type, enhance or suppress the T helper mediated allergic responses. So basically activin is sensing the presence of these allergens, and as we understand, these allergens will display certain patterns, which is where we were saying PAMP and DAMP. Those will be sensed by the system, activin will be made, and activin will activate this signaling process. Also, in case of allergies where skin allergies are there, dendritic cells, these cells permeate into the region where allergens are, and they clear them or mount the allergical responses, the inflammation and so on, by noticing the presence of activin A signaling. So by multiple different routes, the presence of cues activate the activin availability, and that activin binds to its receptor and activates the RSTK, receptor serine threonine kinase signaling through the activin receptor. Further, in cases where you will have inflammation or allergen is present, the cells of granulocyte type, eosinophils and neutrophils, they reach to the location, they release their contents, the granules which are there, and also macrophages are activated in those regions through this activin signaling, and they kind of produce an asthma-like condition, or sometimes they may make the situation worse because of these high activity of eosinophil, neutrophils and macrophages at the location where asthma-like situation is produced. So we realize that activin is again playing very important role in another response which is kind of safeguard for us, and which is your allergy response. So here what will basically happen, whatever is the allergen, that basically is not directly binding to the receptor, rather it is activating the process by which activin A is produced. This activin A binds to its receptor, phosphorylates, goes through the SMAD pathway, activates or inhibits transcription. So remember, when there is a response by default present, the signaling will ensure that inhibition happens so that the situation is under control. Because if you already have a response mounted normally, if this signal activates it further, it creates a situation of a problem or a concern. So depending on the cue, depending on the response which was already present, the system activates or inhibits the transcription of the right set of molecule. So we saw that how RSTK dependent activin signaling is helping in the process of infection and in the process of allergy. Further, what is known that the signaling plays very important role in autoimmune disorders. The pathogenesis or the pathogenic conditions which are seen under autoimmune response. So there, the cells which are actively engaged in responding to the activin signaling are B cells, which we know as the cells which produce antibodies. Then there are monocytes. I already told you, these are the cells which will have contents inside their cytoplasm, which will be released into the extracellular environment, which will affect, clear the pathogen or something which may induce the inflammation. This also involves the microglia. Remember, we said that this is a cell which is present in central nervous system, which helps in the clearing of the infection, as well as the macrophages itself, which are the phagocytic type of cell. So in autoimmune condition like rheumatoid arthritis, the activin signaling is sensed by the macrophages, and this situation is worsened or in further signaling is increased by the presence of macrophage in the system, and the situation is further worsened. More and more rheumatoid-like phenotype would be there if more and more activin signaling is being induced and macrophages are being engaged over there. So multiple different types of signal, multiple different types of cells and different locations, they are mediating different responses. Here the autoimmune condition of rheumatoid arthritis. Or this signaling can activate a pro-inflammatory response through monocytes and macrophages. What I told earlier, that these monocytes kind of cells, they release the content which can induce the inflammation. So when activin signaling is activated by presence of certain indicators, certain cues, certain signals, the monocytes are activated and you end up activating the pro-inflammatory responses. Where here when we are saying that pro-inflammatory responses through gene expression, basically, which means that when the signaling is culminating in the nucleus, you are seeing the expression of pro-inflammatory molecules. And by working through this B cells, which are known to make antibodies, and the activin signaling ensures that autoantibody production is inhibited. We understand that in an autoimmune disorder situation, you will have B cells which will be making antibody against own antigens. So activin signaling recognizes this condition and kind of dampens the activity of B cell. That is why reduced autoantibody is produced. More importantly, in CNS, central nervous system, activin signaling affects oligodendrocytes. These are specialized cell types which do not have immune function, rather they have protective function in the sense that they bind to the axon of the neuron and protect it through myelination. So activin signaling affects oligodendrocytes and it helps in the differentiation and remyelination process if there is a damage. So normally they will prevent any damage to happen, but for any reason, if the damage has happened through this signaling, they will be differentiating further so that they can perform the function of myelination and restore the myelination of the damaged axon. So in some sense, what we saw that activin signaling in response to cues, SMAD, inside the nucleus, I said that it can activate pro-inflammatory genes, or it can ensure that the autoantibody generation is reduced or diminished. So basically here it is activating, and in this case it is inhibiting the autoantibody generation. Now let's look at another situation which often seen with people, where there is a tumorigenic growth and you have uncontrolled growth of cell which can actually lead to the cancer. So here you have T cells, here you have dendritic cells and macrophages which are actually working towards tumorigenesis and immunosuppressive properties. So we know that there are T cells which are involved in mediating the tumor related control. So what has been known for the activin signaling through receptor serine threonine kinase is that T cells infiltrate in cases of melanoma, that reduces when there is a activin signaling. So basically they are promoting the tumor formation or tumor expansion and growth, because this T cell would have reduced the, or would have oncolytic activity, ensure that the tumor does not grow further, but this infiltration is reduced, so activin signaling is adding towards the tumorigenesis process. Further, remember that depending on the cell type, depending on the availability of the cues, the system will respond differently. So here tumor specific T cell responses dampen the tumor growth. So same activin signaling, but a different scenario, you have T cell responses which dampen the tumor growth. In case of melanoma, we see that it is kind of inhibiting the infiltration and probably promoting the tumor growth. In case of again epidermal skin cancer, the activin signaling suppresses proliferation and infiltration of particular cell types. So, it is different cancer types, different tissue origin and different cues, but one of the final output is towards the growth and development of growth of that tissue, growing in number, that is being affected by the activin signaling, and depending on tissue type the response is changing. It is importantly shown that in case of pancreatic cancer, the macrophages are activated and these macrophages now make these antimicrobial peptide secretion. Basically that will also inhibit the cancer growth. So, together what you can understand that activin signaling through its receptor and SMAD phosphorylation inside the nucleus depending on the cell type and cell proximity, because we saw cases of melanoma, skin epidermal cells and pancreatic cells. If we talk about T cells, in all these cases the cell type is same but the cell proximity is very different, and this is where you saw it could be suppressing versus activating the growth. So, this is one cell type or one signaling type which is very critical in proliferation related things, infection related things, immunity related things and so on. Now, we will discuss about another receptor which is of receptor serine threonine kinase type, which responds to this anti-Müllerian hormone. So, just to let you know what is anti-Müllerian hormone, this is basically or let me start with the fact that in a growing fetus, the gonads are bipotential in nature, which means that in presence of right set of cues, they will become a male structure or they will become a female structure. And this is where if you simply look at the growing gonadal structure in the fetus, there are different ducts which are present. If I can put the two ducts in two different colors, so one of the duct which is called Müllerian duct, when this duct differentiates, you get female structures. But when you have other duct which is called Wolffian duct, you will have male structures coming out. So, in case of a situation where a fetus is destined to become male by virtue of having this chromosome Y, which has the genes which are called SRY family of genes, they help in this transition, as well as during the different growth period you have the testosterone levels which work to ensure that the differentiation is towards Wolffian duct type, not towards the Müllerian duct type. So, you can say that your testosterone is working against the Mullerian duct differentiation, and that is mediated by this hormone called anti-Mullerian hormone. And what we have come to know, that this hormone also responds through the receptor serine threonine kinase. So normally you think about signaling which is by receptor serine threonine kinase. You have again the heterotetrameric assembly of the receptor and binding of the AMH activates the SMAD, co-SMAD forms, this moves into the nucleus, and AMH responsive genes are activated. But how this signal is regulated, I told you the AMH is an hormone which will also have a direct component through the testosterone. So in male or female body, there are two different types of hormone, which are luteinizing hormone or follicle stimulating hormone. They work on different cell types in the gonads and affect the levels of testosterone or androgen. That regulates the level of AMH and so on, thus the differentiation. So if you consider a scenario where the signaling is going on normal, anti-Mullerian hormone, so the genes will bring a change which will be towards the male characters. But when there is the signal is not available, there is no signaling. But even when the signal is there, there is a regulation of availability of the signal, and this is where we have discussed this, that how a signal can be produced as a precursor to ensure that although it is available but it is not active and not responding to the receptor. So first this precursor of AMH is processed to make the functional molecule, which needs further processing to make the mature AMH. This is processed AMH, this is precursor AMH, pre-AMH if I write, this is processed AMH, and you have the mature AMH. This mature AMH will bind to the receptor and involve in the signaling. So in many disease conditions, the receptors will be perfectly fine, but the AMH level or its production or its processing will be affected. Sometimes you can think that if the signaling is not happening, what will happen? By default, the system will be set towards the female development. So in the absence of AMH, the process will move towards the female development. So any situation where gonad development is affected, you will have levels of testosterone androgen perturbed, which will affect this anti-Mullerian hormone signaling cascade, and thus will create a confusion towards the development where the genotypically the organism will be male type, but because of the problem in this receptor signaling, the development will be female type. And many such cases are known where whether during the fetal process the hormones were not balanced, or during the puberty the hormones did not come in right quantities, and there were defects in the gonadal development and the signaling process. So what we understood in this class today, that a particular ligand which is activin, which works through the activin receptor, which has multiple role via its signaling activation through this RSTK. It is involved in the infection process or infection response process as well as allergic response process. We have also seen that how activin signaling is contributing towards autoimmune condition or pro-inflammatory conditions and cancer pathways, where promoting cancer or inhibiting cancerous growth. More importantly, we have seen that an hormone which is AMH, anti-Mullerian hormone, or sometimes it is also called Mullerian inhibiting substance, both work through this RSTK and ensure proper gonadal development, male, female. This bipotential nature of gonad takes shape because of the presence of anti-Mullerian hormone signaling. Thank you.